Thank you, Susanna. Thank you, guys, for everyone for having us here. It's an honor, a pleasure to share this knowledge with you. So today we'll be talking about machine learning in life science research and drug discovery. I hope my voice gets along, so you can hear me well, right? Okay.
Yes.
So yes, here is a brief outline for today's talk. So we'll briefly talk about the central dogma of molecular biology. So it's going to be a little bit of introductory information about biology, some important concepts that will be important throughout this talk. Then we'll show you some examples of biological chemical data available. Romeo will show you in details how to work with chemical data, especially share his experience with that. We'll talk about challenges in biology, current challenges, and how AI solutions have been applied so far. And finally, we would like to show you a pipeline from beginning, from a problem to solution pipeline. As an example, our project that we did together at Elix, so to do drug discovery against coronavirus causing COVID-19. And finally, we'll have a discussion. So let's start with some concepts in biology. So it would be important to just briefly mention some certain terms that will be important, especially we'll be hearing about proteins a bit more in this talk. So first of all, nucleotides, just know that they are building blocks of DNA and RNA. I'm sure you have seen them, those ATGCs. Just know that much would be enough. And amino acids, on the other hand, are building blocks of proteins. And what are proteins? Proteins are they are macromolecules like hemoglobin or some other proteins, insulin, you might have heard of them a lot. And they perform quite essential functions in our bodies. That's why they are very much important for our bodies, and as well, they play a lot of roles in the diseases, and they are most of the time targets for therapeutic drugs. And genes in DNA, they encode information for protein production. And very interestingly, there are totally 4 different nucleotides. We're talking about DNA here. So like I don't name them, ATGC, know that much. And finally, they can give rise to 20 different amino acids. And later on, we'll see how it happens. And this whole process from DNA, from gene to protein production is called the central dogma of molecular biology. We have gene encoding a protein and intermediate RNA. Finally, we have a protein produced. To better understand how this happens, I put slide animation here. Unfortunately, the sound of it doesn't go, so I'll try to present throughout this video. So here are genes within the DNA, and then they encode information regarding the proteins. So here we have an enzyme called RNA polymerase. It binds to the gene. So it moves along the DNA, and then to produce messenger RNA, certain, again, nucleotides bind to it, read DNA information, and translate it to, transcribe it to RNA information. We call this transcription. And later on, this RNA moves out of nucleus, but before it, it needs to be cleaved to remove non-essential regions in RNA that were originally in DNA to make just enough information for protein production and nothing extra. And then we move out of nucleus into the cytoplasm. And here, this RNA is later on bound to ribosomes, the machinery inside the cell that produces the proteins. And then ribosome reads the messenger RNA to produce a chain of polypeptides of amino acids that will finally fold into protein. So here's the transfer RNA molecule. It's quite specific to the code of DNA. So each amino acid is encoded by a triplet of code. That's why the combination of those nucleotides can give rise and encode information for different amino acids, which are necessary to be recruited to form certain proteins. And of course, it depends from protein to protein. Everyone has essential their own characteristic and like a fingerprint. Finally, this chain of amino acids is produced, and once it's produced, it folds and makes a protein. So this is we went from gene to protein. Then when we go to working with biological and chemical data, so of course we have those chemical structures, but we need to somehow work with them. And for the simplicity, we write them as text, like ATGC, so that we know what it is. But even though it's text, the meaning behind is chemical information, right? And similar applies to amino acids. Amino acids are also chemical structures. Don't be distracted by the chemistry of it. Just try to focus on the general idea. We don't need that much chemistry here. Just for the sake of showing that there are chemical structures, for the simplicity, we just write them as text and use them as text. But when we work with them, we do not consider them as plain text as in other applications. For example, here is a sequence of amino acids that form essential protein in this COVID-19 causing virus. So we see it as a text, but normally this information actually encodes something like this. It encodes 3D structure of this protein finally, and those helices or sheets are different fragments of these amino acids, and their chemistry actually defines this 3D structure. That makes it quite complex actually to predict 3D structure. We'll talk about it later. And from now on, I'll pass this presentation to Romeo to share with us his experience in working with chemical data, and then I'll take over it again.
Thank you. You should be able to see my screen now, so thank you for having me here. I'll jump right into it as soon as it loads. So before we can train a model, we need to convert the molecular data to a format we can easily work with, a format the models can understand. And this process is sometimes called featureization. So you might be able to encounter the term in some papers. But in general, when it comes to formats, they can generally be grouped like this. So there are discrete formats, which are text-based formats, and some examples include smiles or inchy strings, fingerprints, and there are several types of fingerprints, and maybe bags of bonds or bags of fragments. In terms of continuous representations, we are usually talking about matrices. Well, 2D, sometimes 3D, but the representation is usually in form of a matrix. So electronic density maps, maybe column matrices are some examples. And finally, graph representations have been very popular recently, and they very often outperform classical approaches. But we will get back to this a little bit later. For now, I'd like to go into a bit more detail on some of these representations. And I'll start off with smile strings. Smiles are one of the most popular representations you will find molecules in. Of course, they have advantages and disadvantages. In terms of advantages, I like to say they are very compact data formats. As you can see, it's basically just plain text, and it can be stored easily in databases or CSV files and pretty much any other format you can think of. On the other hand, small strings are not unique, and they do not contain any atom coordinates, so no 3D coordinates. As we can see in this example, which is vanillin, you can write this molecule. So looking at the first example, if we start from this atom, then we see in a parenthesis here, there's a side branch of carbon followed by a double bonded oxygen. Then we continue on the main scaffold with 2 carbon atoms. Then we have another side branch, another carbon atom, another side branch. Finally, we close a ring, which is marked by the one here. And yeah, there we go. We have Small's representation of vanillin. But of course, we started from this atom. We could have just as well started from this atom here, which you can see here. And moving on this side, so we have an oxygen, a carbon atom, and all the rest. Of course, we could just as well have started from any atom from the molecule. What I'm trying to say is small strings are not unique. And also, as I mentioned, in terms of 3D representation, although smiles can handle stereochemistry to some degree, as we can see L-alanine and D-alanine. So using this special symbol, we can sort of tell in which way this branch is pointed to, but that is the limit of smiles when it comes to 3D representations. Now, of course, you could train a recurrent net like LSTM and try to create a generative model which predicts the next character of a smile string. But most of the time, you would probably just end up with an invalid entry. Not to mention, if you try to build a predictive model, smile strings is usually not the best way to do that. The truth is, smile strings are great for storage, but not so much for training machine learning models. For that, fingerprints are a better representation. There are many types of fingerprints, but the main concept is pretty much the same among all of them. So first of all, you split your compounds in subfragments. You could do this, it could be something as simple as splitting the molecule at each individual bond, then maybe after each second bond and after each third bond up to, let's say, up to a distance of 8 bonds. And you end up with a bunch of fragments. Then a hashing algorithm is applied to these fragments, and you get a unique identifier. And these identifiers are then used to build a fixed length binary representation. In the example here, which is using a circular fingerprint, a radius is used. Most commonly a radius of 2 to extract the fragments. So there are advantages again and disadvantages to this approach. So as I mentioned, the fingerprints have a fixed size, usually 2048 bits or 1024 bits, and some of these bits can collide. So we basically have the same problem that we had with smiles, just, well, exactly the opposite problem. Instead of having one molecule with multiple representations, we can have multiple molecules with one representation. But on the other hand, this representation is very friendly in terms of building models. So this string, like each bit represents, well, either the presence or the absence of one of the subgraphs, can be fed to like a simple linear function, support vector machines, random forests, you name it. It's very easy to train a simple model with it. Moving on, let's talk a bit about how we can represent 3D information as well. Column matrices make use of nuclear charges as well as 3D coordinates of the atoms. The matrix is computed using this formula. So as you can see, off-diagonal elements make use of the distance between the pairs of atoms to compute the repulsion between them. And diagonal elements contain information about the self-energy of each atom. So can be seen, we can see from this example here. So this is a carbon atom and an oxygen atom. It's very easy to tell based on the diagonal values that these are hydrogen atoms. And of course, each value here is a mix between the individual energy values and the distances between each pair of atoms. Now, density maps. You can think of density maps as having your own atoms in a 2D or sometimes 3D space. Think of a matrix where the distance between each element is maybe 0.1 angstroms. Then based on the coordinates of each atom, we fill the space with each atom's energy value. We can get maybe something like this in the end. And well, you can run convolutions or pooling operations on this data, but of course, you can already see that we end up with a very sparse representation here. And this isn't ideal in most cases. Some researchers tried approaches like adding a Gaussian blur, maybe performing a wave blur as well, as can be seen here, to reduce the sparsity of the data. And usually this improves performance. And now, a quick introduction to graph representations, which are a very hot topic at the moment. So if you think about an image and how classical convolutions work, each pixel is the same distance away from its neighboring pixels, right? So we are working with the Euclidean space with a fixed distance metric. But this isn't always ideal. If you think of a social media platform, they often use graph databases to store their information. So graphs are made up of vertices and edges, or well, nodes and connections. If you think of social media, a type of node could be a user. And a user can have several attributes like the username, real name, address, date of birth, and so on and so forth. The users can also have friends, which are also like other user nodes in the data set. But besides just individual nodes, their two pairs of nodes can be interconnected with edges, marking, well, that in case of a social media platform, that they are friends. But these edges as well could have additional attributes. So when the friendship was formed, maybe, or the relationship between the two users, maybe they're coworkers or college friends, who knows. When it comes to molecules, it's also very tempting to represent atoms and their features as vertices and the bonds between them as edges. So here we can see an example of the feature representation on the adjacency matrix of aspirin. For the feature vector, for the node features, so to say, so each row represents one atom and each column stands for one feature of that atom. So in the first part here, this could maybe be a one-hot encoding of the symbol. So we can see this could stand for carbon. It actually does stand for carbon. So these are oxygen atoms, and you can, basically these are handcrafted. So it can be anything from hybridization, from how many neighbors each atom has, how many hydrogen atoms it's connected to, whether if it's in a ring or not, its valence, you name it. Then the second component is the adjacency matrix, which describes how each atom is connected to one another. We can see like the first atom here is connected to the second atom, and the second atom is connected to the first one, and the third one, and the fourth one, and so on and so forth. Now, of course, we need to find out what we're trying to do. Are we trying to predict atomic properties? Maybe we're trying to predict bond properties. There's also the case of graph classification, where we might want to predict the property of the whole molecule. So based on what you're trying to do, your models will probably have different architectures. So just as an example, for a node classification, this is a base graph convolutional network, and it's made up of convolutional layers, as can be seen here. Convolutional layers generally aggregate features from neighboring nodes, very similar to classic convolutions, and the central nodes are updated based on the neighbor features. This is done for every individual atom, of course, not only for one. So by adding multiple convolutional layers, information from farther away nodes are brought towards the central node. And in the end, we will end up with features from which include information from the whole graph. Now, if we want to do graph classification, usually we make use of graph pooling layers as well to downsize the graphs. So we can see after we perform a regular convolution, we perform pooling, and we end up with a smaller graph. And in the end, we gather up all the features from the remaining nodes and then pass them through a series of fully connected layers before doing the final classification. And time is a bit short, so I will skip some slides, but I just want to make a final note that base graph convolutional models are not the only options out there. So as we talked the classic approach is to gather the features from neighboring atoms. But if we take a look at the directed acyclic graph, it also aggregates features from further away nodes before the central node is updated. In terms of the weave model, so here the atom features are basically exactly the same as the graph convolutional model, but for connectivity information, features are encoded for all pairs of atoms, not only directly bonded ones. And these bond features are also updated in addition to the node information. For the message passing network, the atomic features are usually passed through bond-dependent neural networks, and the central node is updated using these outputs. So the type of node, the type of bond is also important for these models. Finally, we have some models here that use 3D informations as well. The deep tensor neural network doesn't use bond information per se. Instead, it focuses on the distance between all pairs of atoms. And the NE model, very similarly, it focuses on the distance between pairs of atoms, but it also includes angular information between triplets of atoms. So yeah, things can get pretty complicated depending on what you're trying to do. Finally, I'd just like to make a small note on some useful tools that you will probably be working with if you have molecular data. So usually you will be using RDKit, especially if you work in Python. It can load, well, a lot of molecular formats. And you can calculate basic molecular descriptors with it, manipulate atoms, add atoms, remove bonds. So any manipulation operation based on a single molecule. You can also create fingerprints or check for similarities. You can even simulate reactions and do energy minimization. Of course, if sometimes you want to convert the format of your data, I'd really recommend OpenBabel for converting between different formats. Now, DeepCam is a very popular library, which includes model implementations, mainly in TensorFlow, including graph-based models. I'd recommend checking it out. If you prefer using PyTorch though, there's a library called PyTorch Geometric, which includes operations on most graph-based operations. Yeah. And finally, if you're working with DNA sequences and genomic data, you probably want to check out BioPython. And with that, I'll let Nazim take over again.
In addition to what Romeo presented, if you want to know where you can find that kind of data, here are some databases that might be useful. So NCBI database includes a lot of gene, protein information, sequence information. You can also find some chemical information there. Protein Data Bank is a very useful resource because it contains experimentally confirmed protein 3D structures, which is not easy to get. And it has a huge library of the proteins. PubChem is a collection of chemical information where you can get it. Chembo is also very useful database where you can find drug-like molecules there. And Zinc 15 is a library especially for virtual screening or molecular docking analysis. So from this part on, let's focus on some challenges in biology and medicine and certain applications of AI in those cases. Of course, it's difficult to mention all of them. I'll just focus on some very much important ones. And I guess the most difficult problem at the moment is one of the most difficult problems at the moment is protein 3D structure prediction. As I mentioned earlier in this presentation, proteins contain amino acids, and depending on the chemistry of those amino acids, their bonds, their positions, the folding prediction can get very much difficult. So it's a very long-lasting problem. It's been decades researchers try to address it, still no good solution for it. And the shape of protein really determines protein's function. So we need to know the shape if we want to design good drugs against the protein. it's very much important, and too many possible combinations really complicate this problem. And so far, the only methods out there are like some complex experimental techniques like electron microscopy, crystal generation, then NMR. Like that, those are certain equipment you need to do those experiments, and it's not present everywhere. So it's an expensive and difficult problem. And it's important to understand for us the structure of the protein so that we can infer some information about the function, develop new drugs, maybe design vaccines, design new proteins with certain functions, etc. So what has been done so far? Every 2 years, there is a community-wide experiment called CASP. What they do, they generate in the background a certain number of crystal protein confirmed protein 3D structures, and then they feed, so they open source the protein sequences and invite community to address the 3D structure predictions. And so far, there were different approaches used to it, but in the latest one, which took place in 2018, DeepMind's AlphaFold algorithm actually won the competition. So DeepMind established a very good team, a multidisciplinary team between structural researchers that work on structural biology and also engineers, and then they built an algorithm that can predict structure of the protein from scratch. Why it is important? Because normally when you have a sequence of protein with unknown structure, you first try to find similar proteins so that you can find a template of known structure, and then based on template you try to model, because it's very difficult to do it from scratch. But they were able to do it, and the results were very much impressive. 24 out of 43 structures were scored correctly, and the runner-up only about 3, so there's huge difference as well. But of course there are certain limitations. It's still in infancy, and there's a lot to be done in that field. Additional field that is very challenging at this point is drug discovery field. Why? So it takes a lot of many years to develop a drug and put it to market. 1 decade, sometimes 2 decades, and it costs a lot of money, up to 2.6 billion US dollars for all the procedure, and most of the candidates, they fail during this process, and especially the initial parts of the drug discovery, they take many years to do basic research, some non-clinical studies before you can get very good candidate with which you can proceed to some animal studies and to clinical trials with humans. So it would be very useful if we can speed up this process or save some like resources. And recently, generative models especially were pretty much used, attempted to be used for this problem. And this is one example that I like. This is a generative model generated by one AI startup in Silicon Medicine. So the advantage of this approach was that they cut that generation of potential drug process they fit it into 21 days from the target selection till they got good hits. And then they designed, synthesized, and did initial experiments in 46 days, which is very much impressive. And if this type of contribution keeps going on and developed, so we can be able to speed up the process and maybe also contribute. But of course, it's not the total solution. There are too many things to test after you get a good hit, but it's very much impressive to cut that amount of time and being able to come up with good candidates in this short period of time. These are two very much major examples that I wanted to give. And of course, there are too many applications, and this biomedical field has been pursued very much. And we need a lot of applications in diagnosis, in vaccine design, personalized medicine. So especially for diagnosis, if you don't have right diagnosis, it's difficult to choose treatment options, and level of experience between physicians varies, and you might not have good physicians in certain parts of the world. So, and there's a lot of medical information in terms of scans, pictures, which can be used actually to train models. And indeed, several models have been trained and have good performance. But of course, it's still under development. We also have a problem where patients respond differently to the same treatment actually, and then you need to come up with a good treatment option, and which is challenging. And in this case, IBM's Watson Oncology project, they try to uh, find out the best treatment option for the cancer patients, help out the physicians. This is also one kind of contribution to the field. And additional example is that every year we face this influenza virus issue where we don't know it, but almost 500,000 people die every year out of influenza, and it mutates rapidly. And we need to generate vaccine every year because of the mutations. But there have been efforts to predict mutations in advance to facilitate vaccine design, and that kind of there are too many things to be done. But I'd like to stop with these examples, and we can talk more. And from now on, I would like to move to a case study. So here with Romeo together, we want to show like from the beginning to end how to do a study, because it has a lot of steps, and this will be the flow for it. So we'll talk about selecting a target for drug discovery project, especially collecting data, training, validating, and getting the results and evaluating the results. So let's start with the target selection. So some brief information about SARS-CoV-2. You all heard of it. Just I want to let you know that it is quite big for viruses. It has a very big genome, about 29 kilobases. So, and this SARS protein virus contains of about 26, 29 protein parts. And each one of them can be considered as a candidate for therapy, but of course, certain proteins, they have more essential functions for a virus. So virus actually binds to our cells of our host, in this case us. It goes inside, and once it's inside, a virus uses our cells as a factory to reproduce itself, because it has RNA, this coronavirus, and all the information is stored in RNA. And we need to really find out the best way to block the virus. So several groups try to target several parts of the virus, and a lot of efforts going on. But in this case, particularly, we focused our attention on this particular protein, namely RNA-dependent RNA polymerase. What it means, this enzyme can read DNA, can read RNA and produce RNA. So that's the function of it. The reason we chose it was that there are several reasons to that. Basically, all RNA viruses, almost all RNA viruses, they have it. Otherwise, they cannot reproduce themselves. And so when I say RNA viruses, influenza virus, Ebola virus, SARS virus, there are many viruses, dengue virus that we face. Recent outbreaks have been from RNA viruses mainly. And this protein itself, it has certain structure, which is very much important, because different proteins, they have different structures, but this particular protein has more or less conserved structure between different viruses, which keeps it similar and maybe possible to be attacked not only in one type of virus, but in several viruses. And especially there are certain regions on the proteins which actually do the job. And in this case, those regions have known to be more or less preserved even between several virus families. So that was the reason for us choosing this particular target. And we came up with questions. Is it possible to train an AI model to find out a signature of RDRP, that's the short version for that protein, signature of inhibitors against that protein? And if we can do it, can we use such a model to find new drugs? And especially we focused on drug repurposing, because as I told you a little bit a while ago, then doing drug discovery takes a lot of time. So when you need a quick solution, now researchers try to find FDA approved drugs that have not much toxicity and which can be used safely, but they try to find which drugs can be used against coronavirus instead of generating new drugs. So we also followed similar approach. And to collect the data, we searched PubChem and Campbell database that I talked before, and then we found RDRP inhibitors for different viruses from those databases, and then we established a training set with as much as experimental information that was available. Especially, we tried to get information on how potent the inhibitors are. These are some metrics used in biochemistry to address how potent the drugs are. So we used those drugs. We established a set of inhibitors tested for different viruses, since there's not much information against coronavirus. So we did it from different perspective approach. And then once we established this training set, we split it into 80% to 20% for training and evaluation. And then we had 700 active molecules, 656 inactive molecules, and we had some additional test sets for evaluation. We'll not talk much more about them, but we use them to see how well our model performed. And finally, once we got our results, we tried it on our set of FDA-approved antiviral drugs that are out there to find out which ones can be used for a particular case. And from now on, I'd like to invite Romeo to talk about the results a bit.
Sure. So time's a bit short, so I'll be short myself. So as you can see, we ran both regression and classification models. So just to summarize, what we're trying to do is find drugs that are able to inhibit RDRP or the RDRP protein. So for regression, we're trying to predict the IC50 values, and for classification, we're basically predicting a binary value whether the drug is going to inhibit the protein or not. So as you can see, for regression, from the R2 scores, we see that some models show the decent ability to predict the inhibitory coefficient. So I'm talking mainly about the graph-based model and maybe some simple multi-layer perceptrons. The random forest performed relatively decently. But in terms of the mean square error, we can see that it's quite large, but this isn't surprising given the large standard deviation. So if you take a look at the base stats on the right side, we see that the minimum value is almost non-existent and we have very large values. Still, these values could be better. On the other hand, the classification tasks are performing much better. So in terms of the ROCAUC, we see some models surpassed even 0.9, which is very promising. If Nazim can move to the next slide. Yes, so here for the regression results, we can see that somehow we can sort of make out a line here. In the far right side, there's also like an entry there, half a dot. And so if we, as a summary, on the x-axis is the ground truth and the y-axis are the predicted values. And if we drew a line somewhere maybe around 130, maybe we would be able to select some molecules that are likely to inhibit the protein. But still these values could be better, and we are still looking into other ways to improve these results, including adding 3D information as well. And if you can move on to the next slide again. For the classification tasks, previously we've seen the results for the validation set. As for the test set, which is slightly different from the training and validation set in general, and Nazim can talk more about that if you have questions. We can see the best models, which is random forest and especially the graph-based models. So there is sort of a trade-off. The random forest model has a very high true negative rate, while the true positive is, so it's only able to catch around half of the true positive values, while the graph convolutional network allows more failures to pass, but also catches more true positives. So the predictions of these models were very lowly correlated. So we thought it would be a good idea to build an ensemble model based on these individual models. We were able to slightly improve the results. But again, this data set is slightly different from the training data set, yet still it is able to predict the right values to some degree. It's still an ongoing process. This is what we have so far, and I'll let Nazim continue.
Thank you, Romeo. So this is the last slide here, what we got so far from our results. And basically we had 136 FDA-approved clinical and antiviral drugs in our test set. And out of them, 46 of them were classified as active by the ensemble model. And it was interesting for us to see that out of those 46 classified, so they fell into different categories. And here about 26 drugs, they target either RNA generating enzyme or DNA generating enzyme, because certain functions are still similar between those. And so the model was able to identify the drugs that target those kind of proteins. And in this test set, we had some known RDRP inhibitors for different viruses, and our model, even though it was trained on RDRPs for various viruses, was able to spot 11 out of 15 RDRP inhibitors, which shows that it was able to learn the signature that is needed to inhibit RDRPs. And interestingly, I just want to show you certain candidates that were picked up by our model. Remdesivir recently, it was urgently approved by FDA to be used against COVID, because it's one of the most promising drugs. And interestingly, our model was able to spot that as well. And baloxavir marboxil is also RDRP inhibitor, which is in clinical trials against COVID-19 now. So those two are directly acting on RDRP COVID-19. And here we have another drug which is known more to block viral entry, but still our model spotted that maybe it has that similar signature, and it's also in clinical trials against COVID-19. And we have a cluster of protease inhibitors, and maybe they also might have a shared function with that. So this is the result of our study so far. We are still working, trying on different sets as well. But so far we got this result, and so we tried to show you why we chose certain target, what kind of data we collected, where we collected, how we implemented it, and how we analyzed the results. So maybe it's time for questions.